Hello everyone, welcome to the channel Microbial World. In this video, we are going to discuss about Corynebacterium diphtheriae. Corynebacterium diphtheriae is also known as Klebsiella bacillus. This is because the diphtheria bacillus was first observed and described by Klebs in 1883, but it was first cultivated by Loeffler in 1884. It is a gram-positive bacillus, meaning it retains crystal violet stain during the gram staining procedure. It appears as purple or violet color rods under the microscope. It is the causal agent of diphtheria. Diphtheria is a major cause of illness and death among children, but now it has become a rare disease after the administration of vaccination. Morphology. They appear as straight or slightly curved rods. Sometimes they show a club-shaped appearance, and this is due to irregular swelling at one end. They form granules. Granules are the storehouse of energy. They are called metachromatic granules. They are also called as volutin granules or Babes-Ernst granules. These granules are formed at the pole of the bacilli and therefore they are called polar bodies. Albert's stain and Ponder's stain shows metachromatic granules at the polar regions. These bacilli appear green in Albert's stain along with bluish-black metachromatic granules at the poles. They also exhibit pleomorphism, meaning the bacterial cells show variations in shape and size. This is most commonly seen in older cultures and also under stress conditions. Chinese letter arrangement. So the bacilli are arranged in pairs, palisades or small groups. They often appear at various angles to each other, resembling the letters V or L. This is called Chinese letter or cuneiform arrangement, and it occurs due to the incomplete separation of daughter cells after binary fission. They are non-capsulated, so they do not form capsule. They are non-motile and does not possess flagella. They are non-spore-forming bacterium, which means they does not produce endospores. Three strains of Corynebacterium diphtheriae are gravis, intermedius and metis. The same toxin is produced by all the strains and they are capable of colonizing the throat. Cultural characteristics. Corynebacterium diphtheriae is an aerobe and facultative anaerobe, which means they can grow in the presence and absence of oxygen. Optimum temperature for growth is 37 degrees Celsius and optimum pH is 7.2. On solid agar media, they form smooth, flat and greyish-white colonies. The colonies often have a granular or matte appearance. The size of the colonies can vary, but they typically appear small to moderate in diameter. To enhance the isolation of Corynebacterium diphtheriae from clinical specimens, selective media like tellurite agar or Loeffler's serum slope can be used. These media contain inhibitors that suppress the growth of other bacteria, allowing for the selective growth of Corynebacterium diphtheriae. A unique feature of this bacterium is its ability to reduce tellurite, a compound present in tellurite agar. This results in the formation of grey or black colonies due to the precipitation of tellurium. Transmission. The source of infection is carriers who harbor the organisms in the oropharynx or skin. Human-to-human transmission is through respiratory droplets, secretions, or direct contact with infected cutaneous lesions. Diphtheria toxin. The pathogenesis of Corynebacterium diphtheriae is attributed to the production of diphtheria toxin. Diphtheria toxin is a single polypeptide chain of approximately 535 amino acids. It consists of two main domains, catalytic A domain and receptor binding B domain. Mechanism of action. Colonization and adherence. The bacteria initially colonizes and adheres to the respiratory mucosa, particularly the tonsils, pharynx and larynx. The bacteria has pili and other surface proteins that facilitate its adhesion to the host epithelial cells. Toxin production. Diphtheria toxin is encoded by bacteriophage integrated into bacterial genome. So what are bacteriophages? They are viruses that infects bacteria. So what we need to understand is that all strains of Corynebacterium diphtheriae are not toxigenic. Only the strains that carry bacteriophage encoding diphtheria toxin gene are able to produce toxin. So upon lysogenization of the bacterium by the phage, the toxin gene is transcribed and translated, resulting in the production and secretion of diphtheria toxin. Toxin uptake and action. B domain, that is receptor binding domain of the diphtheria toxin, binds to heparin binding epidermal growth factor precursor on the surface of susceptible cells such as respiratory epithelial cells. So this heparin binding epidermal growth factor precursor is a transmembrane protein, and it plays crucial role in various cellular processes such as cell proliferation, differentiation and migration. So following binding, the toxin is internalized into the cell via receptor mediated endocytosis. Once inside the cell, the A domain is cleaved off and released into the cytoplasm. And then the A domain ADP ribosylates elongation factor 2. So what is ADP ribosylation? It is the transfer of ADP ribose moiety from NAD+, that is nicotinamide adenine dinucleotide, to a specific diphthamide residue in elongation factor 2. Diphthamide residue is a modified histidine residue. So ADP ribosylation of elongation factor 2 inhibits protein synthesis, leading to cell death. Now let's look into the clinical manifestations associated with Corynebacterium diphtheriae infection. Respiratory diphtheria, sore throat. So the initial symptom is often sore throat, which may progress to severe pain and difficulty in swallowing. Pseudomembrane formation. A grayish-white pseudomembrane might develop over the tonsils, pharynx and larynx. This membrane consists of fibrin, dead epithelial cells and bacteria. Airway obstruction. So the pseudomembrane formation can cause airway obstruction, leading to respiratory distress. Hoarseness. Involvement of the larynx can cause hoarseness and voice changes. Cough. A barking cough might be present due to laryngeal involvement. Cutaneous diphtheria. Skin lesions. So cutaneous diphtheria often presents with skin ulcers covered by a greyish membrane. Erythema, which means the surrounding skin may become red and inflamed. Swelling. The regional lymph nodes may become swollen and tender. Systemic complications include myocarditis. Diphtheria toxin can cause myocarditis, which is the inflammation of heart muscle. This can cause arrhythmia, heart failures and other cardiac complications. Neuritis. Neurological complications such as peripheral neuropathy and paralysis, especially of the eye muscles, can result because of the neurotoxic effects of diphtheria toxin. General symptoms include fever. So patients present with low grade and high fever depending on the severity of infection. Malaise. Generalized weakness, fatigue and malaise are also common. Lymph adenopathy, which means swollen and tender lymph nodes, particularly in the neck region, might be present. Laboratory diagnosis. Specimen collection. Clinical specimens are collected from the site of infection, such as throat swabs for respiratory diphtheria and wound swabs for cutaneous diphtheria. For suspected cases of diphtheria, it is essential to collect specimens before initiating antibiotic therapy. This is because antibiotics can inhibit the growth of bacterium and interfere with lab diagnosis. Culture. As we already said, the primary method for isolating Corynebacterium diphtheriae from clinical specimens is to culture on selective or enriched media. So specimens are inoculated onto tellurite agar or Loeffler's medium. The plates are then incubated at 35 to 37 degrees Celsius in a carbon dioxide enriched atmosphere for 24 to 48 hours. Then the bacteria forms smooth greyish white colonies with a granular or matte appearance. Gram staining. Gram staining of isolated colonies is performed to confirm the morphology of the bacterium. Corynebacterium diphtheriae are gram-positive and they appear as purple or violet color rods under the microscope. Biochemical test. Corynebacterium diphtheriae are catalase positive, oxidase negative. It degrades glucose in maltose along with the production of acid but no gas. It cannot ferment mannitol, lactose, trehalose or sucrose. It is hydrogen sulfide positive and reduces nitrate to nitrite. It does not liquefy gelatin nor hydrolyze urea, and also it does not form phosphatase. Toxigenicity testing. ELIC immunodiffusion test. This is used to detect the production of diphtheria toxin by Corynebacterium diphtheriae. So in this test, a strip of filter paper embedded with diphtheria antitoxin is placed perpendicular to a line of isolated colonies on agar. If the colonies produce diphtheria toxin, then precipitin line forms between the colonies and the antitoxin strip. PCR. This is used to detect the toxin gene tox in Corynebacterium diphtheriae isolates. This is a very rapid and sensitive method of toxigenicity testing. Serological test such as diphtheria antitoxin neutralization test is used to confirm the presence of circulating diphtheria toxin in patient serum. So this test involves incubating the patient's serum with a standardized amount of diphtheria toxin and then measuring the residual toxin activity. So a reduction in toxin indicates the presence of neutralizing antibodies against diphtheria toxin. Molecular typing methods include pulsed field gel electrophoresis and multi-locus sequence typing. So this is used for epidemiological studies and also to differentiate between different strains of Corynebacterium diphtheriae. Treatment. Diphtheria antitoxin. So diphtheria antitoxin is administered to neutralize the circulating diphtheria toxin and prevent further tissue damage. The diphtheria antitoxin is given intravenously or intramuscularly. Antibiotic therapy. Antibiotics are used to eradicate the bacterium and reduce the risk of transmission. First line agents used are penicillin and erythromycin. So penicillin G or amoxicillin are the commonly used antibiotics, and for patients who are allergic to penicillin, erythromycin can be used as an alternative. Supportive care. Airway management, cardiac monitoring, nutritional support. So in cases of respiratory diphtheria with airway obstruction, supportive measures such as oxygen therapy, mechanical ventilation and also tracheostomy may be required. And also due to the complications like myocarditis and also other cardiac complications, continuous cardiac monitoring is very essential for patients who are having severe diphtheria. Adequate hydration and nutritional support is also important, especially for patients who are having difficulty in swallowing and feeding. Isolation and infection control measures. So for patients with suspected or confirmed diphtheria, respiratory isolation should be done in order to prevent the spread of the bacteria. And also hand hygiene and other infection control measures are essential in order to prevent the secondary transmission of the disease in healthcare settings. Vaccination. Diphtheria toxoid containing vaccines. DPT vaccine, that is diphtheria pertussis tetanus vaccine. So in children, the diphtheria toxoid is given as a trivalent preparation. And the adult type vaccine is DT vaccine, that is diphtheria tetanus toxoid. Okay, so this is all about Corynebacterium diphtheriae. I hope that the video was helpful for everyone. Do subscribe the channel for more informative videos. Thank you.